Green plants get their energy from the sun. Within their cells, light energy is used to synthesize proteins, fats, nucleic acids, and carbohydrates, the molecules of life. This process, photosynthesis, occurs in chloroplasts, the rounded green bodies found in leaf cells. Elodea leaves are made up of just 2 cell layers, offering an unobstructed view of living chloroplasts. The orientation of the flowing cytoplasm determines the amount of light hitting each chloroplast. When light levels are low, the flow is adjusted so that the chloroplasts get full exposure. Under the more intense light of midday, they circulate around the sides of the cell, reducing exposure to harmful ultraviolet radiation while still receiving plenty of light for photosynthesis. The chloroplast's internal structure was recorded by the first electron microscopes. They revealed that a chloroplast consists of an outer membrane, an inner membrane, and stacks of hollow discs called thylakoids. Lining the thylakoid membrane, a carpet of light-trapping chlorophyll molecules. The soup of enzymes surrounding the thylakoid discs and contained by the inner membrane is called the stroma. This is where the molecules of life are synthesized. Chlorophyll absorbs light energy in the red and blue regions of the spectrum. Green is reflected away, which explains the color of most plants. When a chlorophyll molecule absorbs a photon of light, the photon's energy transfers to an electron. These energized electrons travel to a reaction center chlorophyll, where they transfer to electron transport proteins embedded in the thylakoid membrane, beginning a process that yields high-energy compounds used in photosynthesis. Another reaction occurs on the thylakoid membrane. Water, H2O, is split into hydrogen ions and oxygen. 2 oxygen atoms join to form O2 gas, given off as a waste product. The hydrogen ions are pumped into the thylakoid space. An enzyme provides an exit. As the hydrogen ions stream out through ATP synthase, energy from the outflow is used to synthesize ATP from ADP. ATP is life's universal energy carrier, supplying energy to drive biological chemistry in everything from bacteria to mammals. With a further input of energized electrons from another reaction center chlorophyll, NADPH, another powerful energy carrier, is generated. The depleted electrons return to the chlorophyll molecules ready to be excited by another photon of light. The ATP and NADPH generated by these light-dependent reactions provide energy for the light-independent reactions that take place in the stroma, using carbon dioxide as raw material. A key product molecule produced in this cycle of reactions is phosphoglyceraldehyde, PGAL for short. The cycle can continue as long as carbon dioxide and the energy carriers made in the light-dependent reactions are present. The output is a steady stream of PGALs. An enzyme converts 2 molecules of PGAL into a molecule of glucose sugar, a basic cell fuel. For long-term storage, plants convert glucose into starch, a storage product that is taken advantage of by animals of many kinds. Green leaves are not the only place to find chloroplasts. Many kinds of single-cell protists contain them, hundreds in Euglena spirogyra, 1 in Chlamydomonas. Multi-cell photosynthetic organisms probably evolved from single cells like these, by way of colonial forms like these. The largest colonies contain thousands of cells, some specialized for locomotion, and others for asexual and sexual reproduction. Such complex colonies were an evolutionary stepping stone to simple multicellular plants.